right, this here is a model, it's mitosis board, so it's showing us the different stages of mitosis. And so this guy right here, if we go over, this is a cell, right? So we have the plasma membrane on the outside, number one, and then inside two is the cytoplasm over here, three, this is the nucleus, and four would be the nuclear envelope. Over here, this green guy in here, which is labeled 5, is going to be the nucleolus. And then within here, we can see these guys that are labeled 8. These are the chromosomes, and these have been condensed. You can see them now that they are chromosomes, they're condensed into chromosomes. Then we also have 6 is going to be the centrioles over here, and they're surrounded by these things called star asters, which is 7. And so these centrioles have already been replicated, and this happened at the end of G2 in interphase, and so now we have a pair of centrioles over here, centrosomes. So because our chromatin has already condensed into chromosomes, we know that we are now in mitosis. All right, so we don't have, this is not interphase anymore. Once the chromosomes condense, it's going to be prophase. Right, so this is going to be prophase, and it's kind of the earlier prophase here. Next, we're going to move over here, and so our centrosomes are moving to opposite poles. Remember, a centrosome has 2 centrioles in it, and they're moving to opposite poles of the cell, opposite sides. This part right here is the spindle fiber. It's labeled 9 over here. These are mitotic spindle. It's made of spindle fibers. And then we have you can see these chromosomes are now even more organized, and you can see that they are arranged as sister chromatids. So they are replicated because remember the DNA replication happened in the S phase of interphase. So this is also going to be prophase here. Just a little bit later in prophase. You can see we still have the nuclear envelope present. Since we still have that nuclear envelope present, it is still going to be part of prophase. Let's move to 3. We can see now that the nuclear envelope is starting to dissolve. We have our little chromosomes here, which are composed of sister chromatids that are attached to the centromere. They are all going to they're trying to attach to these spindle fibers here, and they are working their way towards lining up in the middle of the cell, but they're not quite there yet. So this is going to be, you can call it late prophase. A lot of books have named this part over here pro-metaphase, right? So this is the part right in front of metaphase, which kind of means early metaphase. So pro-metaphase or late prophase. I'll accept either of those. 4, though, is going to be definitely metaphase over here. So metaphase, we know that metaphase has happened when all of our chromosomes are lined up down the center of the cell. We can call this the equatorial plate or the metaphase plate. We can see that our sister chromatids are still attached right here at that centromere in the middle, so it's still metaphase. 5, the main difference between 4 and 5 is that now we see that our sister chromatids are not attached anymore. They have been pulled apart. They're being pulled away to opposite sides of the cell, and so this one is going to be like an early anaphase. So this one's anaphase because our sister chromatids are not attached anymore. They're not attached at the centromere like they were before. So they're being pulled apart. And now we can see 6 over here. This is going to be a later anaphase because our sister chromatids are now being pulled to either side, but we have not reformed the nuclear envelope yet. When we get to 7, we notice that our nuclear envelope has been reformed, our mitotic spindle has broken down, and so this is going to be telophase. Other things that are happening now, all of the chromosomes are they're on either side of the cell. We have our nucleolus reforming during the end of telophase, our nuclear envelope is reforming. And now, this started during the end of anaphase, but we can see here that this is the cleavage furrow. And this cleavage furrow is going to be the beginning of cytokinesis. So mitosis is going to be what happens in the nucleus, whereas cytokinesis is going to be the division of the cytoplasm in the organelles. And so this is going to be the cleavage furrow at the beginning of cytokinesis. The cleavage furrow deepens here. So it starts a lot of times in the end of anaphase. It continues throughout telophase, and then after telophase is over, it eventually finishes. And then we have two resulting cells at the end, and those are called the two daughter cells. So each daughter cell should have an identical copy of chromosomes. We should have one centrosome in either cell. And at this point, we can see that these cells are back in interphase because the chromosomes are no longer condensed. So we can't see those condensed chromosome figures anymore, so we say that these daughter cells are now in interphase. So they can then go through the phases of interphase and eventually go through mitosis again. They're going to continue dividing. So this part right here would be interphase. And these are two daughter cells.